Hello again. So as you can see from the title, and as Roberta was saying, this is something very, very different compared to the first lecture I gave. I tried to motivate why, in the beginning of the first lecture, why the selection of this. It was very inspired by the book that we wrote. And so this will be, to some extent, a lecture, meaning I will introduce things, but I will move a lot to model systems that we work with to show you basically what you can do in terms of bio-inspired design, because that's the simplest way to show you what is really possible. If at any point anything is unclear and if I'm going too fast, please stop me and ask questions, because it will be less formal and I will do more hand wavy things. So if it's not clear, please interrupt me. Okay, hopefully this works. Or doesn't? Yeah. Okay. So we are all made of star stuff. This is one of the most famous sayings of Carl Sagan. If you know who Carl Sagan is, you can consider yourself not young. If you don't know, you should definitely check it out. But or that, thank you. or you forget. Thank you, Luca. But so when many of us were growing up, this was one of the most famous educational series about cosmos. And so this is what basically Carl Sagan said. And what this means is really summarized in this picture over here. Very basically, this is a zoom into an Orion nebula from it was a Herschel Space Telescope, where scientists did analysis of the spectrum of what is seen here, and what they found is ionized carbon atoms. Or in other words, what this summarizes is that some 4.5 billion years ago, all these heavy atoms were really created in first generations of stars, okay? And this is what we are made of. And so it's sort of a nice sentiment, and reiterated in a slightly more chemistry-oriented way. Life, as we know, it is very complex. It has lots of variations, but if you boil it down, the most basic elements out there, it is all this star stuff. So hydrogen, carbon, oxygen, nitrogen, sulfur, and phosphorus. And then if you look at really at the nucleic acids, RNA, DNA, they are comprised out of all of these elements. Some amino acids and proteins go up to sulfur. Others, amino acids and proteins, again, go to nitrogen. Carbohydrates and lipids up to oxygen. And of course, hydrocarbons are just hydrogen and carbon bound together. So these few building blocks do make this plethora of life here, this tree, little echidna, leafy sea dragon, dragonfly, and many are. So life forms very broad and very diverse, comprised basically just out of few building blocks. Okay. Now, so they're broad and diverse, but they're really dominated by accidents and environmental inputs that were good at a given time when these things were being formed and evolved. So one of the central questions for researchers coming from various fields, physics, chemistry, biology, engineering, who are interested in life around us, is basically what are the laws and rules behind what we see in nature. As I was trying to sort of hint by writing this, life as we know it is just a possibility, okay? And in the 90s, Christopher Langton wrote, to have a theory of actual, it is really necessary to understand what is possible. Or in other words, life as you know it is one instance of things, but to really understand how it works, we need to understand what are the possibilities around it. And then maybe we can actually have theories of what we see in nature. And that sort of spurred the field of artificial life, and in many ways, bio-inspired design. Basically, because people started looking into how you can take building blocks not completely just limited by what we see in nature and the chemistry that we know, but any kind of a building block, how we can endow it with information that will then lead it to building objects that have some properties that are maybe bio-inspired, but maybe something else. I hear some programmed function, things can transform, self-replicate, all with the goal of trying to maybe discover new materials and new properties that can be interesting for us. And by new properties, an example would be for this one here, where by manipulating the DNA, coated colloids, which I will go into more details in a little bit, what Ben and Winnie were able to find, to figure out, is how they can make a crystal at intermediate temperatures. So at low temperatures, it's a gas. At high temperatures, it's a gas. But at intermediate temperatures, it's a solid. We can discover new things, but you can also learn about biology. So this is the work from Hod Lipson. At the time, he was at Cornell. Now he's at Columbia, where he was, again, taking building blocks that have arbitrary options and functions. And the only thing that he wanted to know is, by using some genetic evolutionary algorithms, how this blob of objects can be evolved to move. And then the solutions that come out are really very much like the ones that we see in nature. So by doing that, he was able to categorize types of motions that immediately come out if you just pose a question: if I have this set of building blocks, how can I make them move? So there are many soft matter systems that people use that are biomimetic, right? That people use to try to mimic what we see in biology. They go over a wide range of scales. So on larger scales, again, these are just examples. There are many more. Here we have an actual, centimeter size network, elastic network that has topology and bonds between these struts designed in such a way that it has what is known as allosteric behavior. In biology, what is known where you have protein complexes. If something attaches here, something else happens far away. So it's a network that mimics that behavior. Here on a very similar scale, what you have are networks that by just shaking using gravity, these little circles are magnets with different interactions. They can be, basically what Itai and Paul do, they shake them, and basically what they do, these networks basically fold into complexes, protein-like complexes. So they try to understand what are the minimal ingredients for this to happen. Here on a slightly smaller scale, we have gels. It's a mixture of gels and DNA, and then by tuning DNA interactions, which I will explain in a bit in more detail, basically what they can do in Pening's lab, they can make them assemble into predefined structures that then have certain properties. Slightly smaller scale, and this is the scale that I will focus on mostly, we again have a few interesting examples of these biomimetic systems. So here in Stefano Sakana's lab, what they make shells with certain protrusions that can basically act as cell mimics. So they can accept material from the outside, they can eject it, depending on some design of interactions between different building blocks in the system. Here we have chains of droplets, now emulsion droplets, that are coated with DNA that can be made to fold into more complex structures. These are DNA constructs in Henrik Zitz and Seth Frieden's lab. What they do is they make these different triangular facets and they make them assemble into virus-like shells, and they're capable, by functionalizing things inside, they are capable of trapping cargo as medical applications. And here we have an example from Paul Chaikin's lab where they can make a very simple replicator, so a system that self-replicates. It's a set of so-called DNA tiles, and then again, by designing interactions between building blocks, and cycling temperature and light, they can actually make them replicate. So what I'm going to tell you about is basically this scale. And I will talk about basically this. So we're going to talk about, in general, how stuff is made in terms of how we make stuff, people make stuff, and how nature does. We're going to talk about some limitations of what we make and how nature does it, and how we can actually approach what nature does more. And then I'm going to talk about something we call supracolloidal assembly and a little bit about self-replication. Okay, so how stuff is made. The way that we make things and the way nature does it, very, very different. So these are sort of like top of the line examples. So this is a computer chip, clearly. We know that it's one of the most complex things a man can make. And this is a small subunit of the ribosome, so it's involved in protein synthesis. So both very complex, made out of many components. So here we have the circuit board, you have different transistor chips, etc. And here for the ribosome, what you have is ribosomal RNA, which is the largest part, and then you have proteins that are made out of amino acids. Now, to get both of these things, you need sort of a blueprint kind of a thing. I mean, of course, it's clear completely in the case of the circuit board, the robot that assembles it, right? It needs instructions where things go, otherwise it will never happen. The same goes here. If you look at this ribosomal RNA, these red parts mark places where different proteins actually bind. So they have many resemblances, but the main thing that distinguishes them is the way that they are made, right? So the chip one, we make it using a sort of what is known as top-down approach, right? We have the components and then we arrange them one by one in following the instructions. Whereas what nature does, nature uses what is called the self-assembly approach. Things are just mixed in a solution, and you just watch, and these things come together spontaneously without any external input, and they form these structures. So in many ways, people have been very much interested to mimic these kinds of things on an industrial scale because it's much cheaper, it's much easier. Just put in components and they do the job by themselves. You don't need any kind of external input. But not only that, the nature-made things can also repair themselves, can replicate, can adapt, can all have many more degrees of freedom than the ones that we make. And so a lot of work in the last 2 decades went to trying to understand this process of self-assembly, because this is the key process in nature. And what will become very clear very soon, if you already don't know, is that these specific interactions are a crucial ingredient in the whole story. Okay, so self-assembly, a bottom-up approach, a process where there's some pre-existing components spontaneously come together and form structures with high fidelity and high yield. High fidelity means no errors. High yield means successfully many copies when needed. Now, if you have a set of these building blocks, whatever they are, you can imagine that you make sort of an ESI set of larger scale objects, right? But if all these building blocks are the same, right, if they're identical, then this is not the only thing that can happen, right? You can have a bunch of other things, including some crystalline structure that can just aggregate and form. So there is no control to get only this. So there is really not information in our system that tells it to assemble only this what we want and not something else. So specific interactions, who interacts with who and how, are the crucial ingredient of this whole self-assembly story. Which then leads us to this broader field of what is called artificial chemistries. So basically, moving away from our traditional chemistry and building blocks that can have whatever interactions we actually want. Things to vary are isotopricity of the interactions, valence, and basically interaction specificity. And one of the main ingredients that people use is DNA. As you know, if you take a strand of DNA, it's to bind to another strand, to make a double helix, to bind completely, you have to have a complementary strand, right? And so if you have a strand of length 10, that basically means that you have over 1 million different types of interactions you can design by just varying the sequence of the DNA strand. So it's very easy to use DNA as what is known as specific glue or as a building block. So as a specific glue, what you basically do, you take these single strands of DNA and you put it on surface of objects. It can be particles, it can be droplets, it can be whatever you wish. And basically two objects, when they come together, if they're coated with DNA, they will bind only if they have complementary strands. If they don't have them, they will not bind. It can be used as a glue, but it can also be used as a building block. So here, let me go first through the example because it will be clearer. So here on the top, this is what you see. These are emulsion droplets. They're a few micron size that are coated with DNA because they are emulsions. So there is a lipid bilayer at the top. When you coat them with DNA, this DNA is basically mobile on the surface of the particles. And so this is what's basically seen here. So they coat these emulsions with DNA, and when two come together that have complementary strands, you have your DNA strands basically condense into binding patches. Okay? These are also emulsions, but here the binding patch is not visualized, just the different types of particles, meaning containing different types of DNA on the surface. These are silica particles, again, coated with DNA, and then there are 7 different colors. Here, each particle has different flavor, okay, in terms of, in the language of these artificial chemistry, these are different artificial atoms that can make a molecule, artificial molecule. Now, if we just look at the purely DNA, what people do is make constructs with it, either very long strands of a virus, and then put small pieces that then glue on specific places and by design make certain objects. Or they can even just use small strands of DNA and then have thousands of them form different structures. Again, I will focus mostly on things that are interaction intermediated by DNA, but this is not the only way to make specific interactions. By just designing the shape of a particle, you can achieve also similar things. Here are a few reviews. Many people work on it. It's not an exhaustive list. Just to give you an idea, these are some names. Okay, so back to DNA. One of the most important thing about these DNA constructs when it's used as a specific glue is that there is something that's called the melting temperature. Basically means there is a temperature below which, if two particles come together that have complementary strands, they will bind and they will stay bound. And above which, they will just separate and melt. This is, so for people like me, a theorist, a simulator, this is like a heaven, which basically, they basically experimentalists tell me, you can have any kind of building block you want, you can have any kind of interaction you want. So for me, that work with spherical particles coated with DNA, which for me, this only means, okay, so I can have any colors that I want, any kind of interactions that I want. And so all my quests start by assuming that all these experiments work very well, which they do. Another thing that is important, and so I hinted it sort of when I was talking about these emulsion droplets, is this, what is called species valence. Again, So you can make anisotropic particles, and that definitely in the assembly process can help a lot if you have pre-designed angles where things should meet. I prefer to work with something that looks more like it's an atom with electrons around, meaning that if you work with emulsion droplets, actually it can also be made with silica particles, but with emulsions it's easier because you have this lipid bilayer. When you put your DNA on top, it can migrate, move on the surface. Also, and then if two come together, they form a patch. These patches have typical size. Basically means by controlling the density of DNA on the surface of the particles, you can control the valence of these particles. And so this is what's sort of illustrated here. We have two types of particles, a yellow one and a red one. The yellow one has enough DNA to bind two red ones, and the red ones have enough DNA only to bind one yellow one. And so this is, this comes from experiments, right? So this is not just my imagination. This works really well. So there is thermodynamic control of valence, and this is a nice paper that talks about it. And so these are usually the two ingredients that I, as a theorist, use when trying to understand how we can mimic processes from biology. Okay, so we talked about how stuff is made, and we talked about artificial chemistries. And so now let's just try to build things. Yes? No. Nothing. Nothing. Absolutely nothing. Nothing. That's really the beauty of it, and this is why it mimics, for me at least, the most how atoms and molecules work. And so you can have 2, 3, 5 different species mixed, and then once the two particles with complementary strands come together, the migration will start of this DNA because they will want to bind. It's lower energy, so they will just want to bind. And to make a patch. So it doesn't matter where they are, they will just eventually come to the patch. More questions? Okay, so let's just try to build things. So how do we build things with these artificial atoms? Okay, back to this story. So we have pre-existing components that spontaneously come together. So what we work a lot in simulations and theory are just colloidal particles, usually micron-sized, so small enough that we have nice Brownian motion. If there is no DNA on top, they will actually, if you just put them in a solution, they will come together and be held by depletion interactions. For this setup of 1 micron, these are the typical at room temperature interaction strength and range. So if you take a lot of colloidal particles, they will just, and you put them in a solution, they will make a crystal, right? But we don't want to work with crystals, right? So we want to make specific objects like nature does. So what we do, we simply go to smaller sizes. So we don't look at thousands of particles, we just look at the few. And the beauty of looking at the few particles is that they're really not like crystals. So if you take 8 particles in 3 dimensions, they will, and you put them in a solution, they will come together and assemble a range of structures. And most of them don't have typical crystal symmetries. They will have some other symmetries. So they really can be quite diverse. Now, another beauty of going to smaller sizes is that you can work with mathematicians who have been enumerating what are known as sphere packings for decades. Incredibly hard problem, but they can push 3D to 14, 2D they can go to up much more, which basically means you know what are all the possible structures that can be formed up to a certain size. And so this makes our life easier. I will explain why in a second, but this is just, if this hopefully works, this is just from an experiment, a case of 6 identical particles in just a solution. I don't know, I find it always mesmerizing to watch them do stuff. So here they're just moving between these 2 different states that are possible, and then it equals 6, but it's still beautiful to see. Okay, so going back to our ribosome example. So if you want to build something like this, and let's say we have 8 particles, what mathematicians tell us is that there are 13 different objects that can be assembled if our particles are identical. But we want just one specific one. So we know that we have to put in some information in our system. We have to make our building blocks specific. So the simplest way to do it is to just take the structure and make every building block in there a different type. Okay? So if I here have 8 particles, so I'm just going to have 8 different types of particles that follow this, let's say, artificial chemistry, these sets of interaction rules. So 8 particles, you like only your neighbor and no one else. So gray means you like each other, white means you don't like each other. And if I take 8 particles that follow these interaction rules and I put them back in the petri dish that I showed you, I will assemble this structure and none of the other ones that are here possible out of 8. And so this is known as an interaction matrix, and this will be a reappearing thing throughout the talk. Okay, now I gave you an example of what you can do. In truth, there are many solutions, often, for a given geometry, for a given structure. And this is what is known as solving the inverse. Yes, please.
The structure you show here has an obvious symmetry, and so it looks like you could reduce the number of interactions.
Absolutely. Exactly. So I explained that in just a second, but exactly you can. So this was just an example. This is sort of thing that works almost all the time. If you have a geometry, it doesn't matter how big it is. If you say everyone is different, you will assemble that geometry with very high yield and nothing else. But indeed, there are some symmetries in structures, and clearly it suggests that other solutions are possible. So the way that we get other solutions is by solving this inverse, what is called an inverse statistical mechanics problem, where you have a set of solutions and you ask, what are the interactions that we need to make only one of them the minimum of the free energy landscape, and not the other ones. You want to make a gap sort of between the one that you want and all the other ones. And so these are just now examples of, this is just for 6. For 6, there are 2 different structures, geometries that can be made with 6 particles that are distinct. And there are 2 different interaction matrices. But here, for example, it's a very symmetric structure. I don't know how much you can see, but in one case, we can make it with 2 or with 8 different colors. There are some structures here with 9 that you can only make with just 3 colors, variation of interactions. And many people have been working and work on this inverse stat mech. So Anna, this answers your question. So indeed, you can have many different solutions. These things that you see here is what we call alphabets. And this is just up to 9 that I'm showing you for 3 dimensions. There are some structures that are highly designable, and they have 200 different solutions, 200 different matrices that code only for them and no one else. And then you have some which are really, really hard to code. They have only one solution. And this already, just at this level, this gives you a little bit of a flavor of what we know from biology. There are some things and some structures that are abundant. You see them in all species, but there are others which you just do not. They're very rare or are very species dependent. So we already have this flavor just at this level, where we still didn't do anything much except design assembly and specifically geometry. Okay. So now, of course, a natural question to ask is, how well do these different designs work for a given geometry? And again, this is just an example. This is for 8 particles, a specific structure. This is a range of solutions that one can find. And here what you see are yields. So this is yield. So basically the success, if I have 1,000 simulations running at a given temperature, how many of them are successful, made exactly this structure at the moment when I look in my system. And so these are the solutions. And so what you can see is that the one where you have 8 different colors, the one that I also told you is basically almost always a solution that you will have, is usually the one that performs the best. Others perform the worst. So why do I say this? So I'm focusing, so high temperature, you don't see anything in the yield because it's really gas, it's above the melting temperature of the DNA. All the simulations that were done here are calibrated to experiments, so all the ingredients that we put in, bond strengths, so solvent, everything goes from the experiment, so they're calibrated to it. So what you have here, it's a gas, so they're not binding, it's not, the temperature is too high. At very low temperatures, they're usually stuck, and so there's some intermediate range where you have sort of an equilibrium result. And if you do a theoretical calculation, what you should expect they match. So this one here at the top, where everyone is different, performs the best, and then the rest are close or doing much worse. But of course, you can try to make it bigger. So what happens when you make bigger structures? Because again, the small subunit of ribosome that I showed you, this is just a small subunit. That one has like 70 components. It's not huge, it's not thousands, but it's not 10, it's much more. And then if you do a plot like this, which is sort of the plot to this maximum yield as a function of system size, you see this sort of a trend going down in the yield. So if I go from 10 to 1,000, it doesn't seem like I can actually assemble much. So one can, of course, now ask the question, so what is really competing with me? Because my design is supposed to select only one structure, the one that I want, and nothing else. So what is really impeding my yield? And do we ever expect to see something on a larger scale, successfully assembled in the lab?
Sorry?
Yes?
What about the time scale of assembly? So here you look at the absolute yield, like over always the same time in simulation.
Yeah. So if you, of course, if you look at shorter times, you might, even though the temperature is, let's say, equilibrium temperature, whatever this means, you will probably sample more things that are at lower temperatures because they will not be completely assembled and they will be stuck. So we wait for long enough.
But that also changes with the number of particles.
Changes, but if you, the thing is that, of course, in equilibrium we can also do a calculation, so we know exactly what to expect, and we also do both ensemble and time average simulations, and that helps us pinpoint exactly where we should look. Okay, so what are the competing states? So what is messing up our yield? Well, so this is sort of summarized in this plot here, and again, this is also will be a repeating feature throughout the talk. This is a potential energy landscape. So it's not a free energy landscape, it's a potential energy landscape. So basically, what you have here is a number of bonds increasing in a given structure. And so here at the top, so this is a a system of 6 particles with 3 different colors. And then what you see here at the top are all the possible structures that you can make with 6 particles bound by 5 bonds. So in the assembly process, okay, if you look at how things assemble, these are all the possibilities, geometrically, all the possibilities that you can have with 5 bonds. And then every next row is a structure that you get by adding 1 more bond from the previous one. So of course, as you go down, the number of structure decreases because in the end, the design was such that you can get only 1 structure. Lines connect geometries that you can make one from the other by either making or breaking a bond. And red means, so blue means if you end up on a blue path, that however you make your next bonds, you will always end up in the design ground state. So if I start from this one, and I made this one particular bond to make a central triangle here, it doesn't matter how the rest of the bonds are made, I will always make the one structure I designed for. But if I'm on a red path, that means that I can end up in something that's called the local minimum. And yesterday you heard a little bit about local minima as well. So it's the same spirit. It's a higher energy state that is a trap for the system. So if the system ends up here, it cannot directly go to the desired structure. You have to break a bond or bonds. You have to go up to be able to go down again. So there is a barrier to cross the ground state. So this is really a competing state, right? So before, when we had identical particles, you were only competing against the different geometries that have the same number, similar number of bonds. Whereas here, you designed for only one, but you can end up in local traps. And of course, naturally, as n grows, the number of local minima also grows. This is just an example for 8. They become completely, you cannot even plot them if you go bigger. So I'm just showing you here one more example. And then here, what is colored are again these what we call unique paths. So if you end up in a certain colored path, you will always end up in a geometry that is circle. So red ones go to the ground state, the blue ones will go to the local minimum. And the gray ones, you never know. You can end up anywhere in the tree. Okay. So we sort of understand what are the competing states, but what does it mean? So does it mean that we can never assemble things? We know from nature that it's possible. It should be possible to assemble things very reliably, with very high yield, very efficiently. But also, there were a series of experiments done in Penkin's lab where they took small strands of DNA, many good thousands of them, and just by designing sequences, they put them in a solution. There was some protocol, but I think it's irrelevant for the story. They were able to assemble arbitrary objects that have thousands of building blocks in them. And the yields were not negligible. They were measurable. You could clearly, wherever they looked, they could clearly see their structures. So it was not a zero yield. Right? So we were trying to understand really what is happening. And then the main conclusion is summarized in this plot. So there are several, so let's just focus on the first one to start with. So what I'm plotting here is the number of local minima as a function of their energy for a structure that has roughly 70 building blocks. And so the number of local minima grows, and of course it grows exponentially with the energy. But what I plot in red here is the yield of the structure as a function of these local minima. And basically what this plot summarizes is that the only thing that matters are the low energy local minima. So they are the ones that are the most detrimental for the success of the assembly. And all the other ones that you can have as your structure grows, because they grow exponentially, they're so expensive, they don't matter. And they're also, if something happens, it's very, very high in the assembly, early in the assembly process, early in the, so it actually really does not matter. And so even though you have here structures, this is just 19, where you have a few, so you have not a lot of local minima, but they're all low energy. They just kill the yield completely because it's easy for them to form. If you're a large structure, the largest number of your minima are very, very, very costly, so you actually don't see them. So you can actually build structures reliably, efficiently, with a little bit high yield. The main thing is just to figure out how you can prune the local minima, and that you can do an experiment. The way that they do it, they have some temperature protocols that they go a little bit up and down in temperature, and they increase the yield. Okay, so we sort of know now a little bit of how we can assemble structures if we start from a gas. So the main recipe is draw your structure. Just say if it's made out of identical, if it's made out of spheres, just take every sphere to be of a different type. That will give you the recipe for who should interact with who. That will work. If it's a large structure and there is some symmetry, you can be clever to figure out fewer ingredients than the maximum number that you will no longer write. But for everything else, if you want something, a smaller number of building blocks, you have to do more work to figure out how to do it. And of course, if the structures are huge, it's really not, it's much more complicated. And so, of course, if we go back to nature, nature doesn't do things just from assembly from gas. There's something else involved there. And so, this is what I was saying, so we can make these things, but you need usually to have a reliable assembly, you need a large set of different building blocks, or, which I didn't talk about at all, but hinted with these different experimental examples, you need very complex building blocks in terms of shape and valence control. But of course, nature just has 20, some says 20 types of building blocks, it's 20 types of amino acids, but the way that it does things is that it chains it, right, into a specific sequence, and then the sequence maps and follows into a three-dimensional structure with a given function, right? So it is assembly, but in a different way. You first assemble a chain in a given sequence, so there is some information that is already coded there, and then you do the assembly. And so we started looking into trying to understand, so how can we do that now in these artificial systems? We moved to 2D because the time that our experimental friends were doing 2D stuff. And so this is again from what I had before, molecules in 2D, that mathematicians can enumerate. This is just the number of these geometries as a function of system size. So you should just look at this lower one, so it's log linear plus, so the number grows exponentially. And then we have the same story that we had before. We have a potential energy landscape. So now we just don't start from the whole gas and all possible states that you can imagine from the gas. Our starting point is a chain, but you can still have a potential energy tree. Again, each row counts different geometries that can be made, starting from a state before. And now, if you go through these in steps through these different rows until the end points, this comprises a folding pathway, right? Because now it's not just assembly, but we are starting from a chain, so it's a folding pathway. And so this is example with seven particles. There are 4 different
geometries that actually now don't have the same number of bonds between them. So this flower-like structure sits here at the bottom, and these 3D geometries sit here already. And so already, if you look at identical particles equilibrium, there is a preferred minimum, and other compact geometries that you can see are just one bond or less. Now you may wonder why there are multiple points here when I say I have only one geometry. Well, this is not because, okay, so we're starting from a chain, but how this chain is arranged in a given structure actually matters. So this is known as a Hamiltonian path, and so how they are arranged. So multiple points here actually just count different ways in which you can arrange these chains into a given geometry, so how they can fold. And so you can see, okay, so maybe we will gain something by doing things like nature. Okay, so we're starting from a chain, so maybe we will be able to do things with fewer building blocks, but there is a whole lot of nightmare coming from just trying to figure out how these things can be arranged. Okay, but that's the basic setup. So we start from a chain, and we say, okay, so we want to build structures, but now not with one building block, but what is larger than 1 or 2. Let's say we have 2 types of building blocks, which means we have 3 interactions, blue, blue, yellow, yellow, and blue, yellow. And what I will talk for the moment are just alternating sequences, because this was what they had in the experiments. And then, but I will generalize later for if it's not an alternate sequence. And so a very important point again is this temperature control. And so this is also, again, a control knob in all these experiments where you can design these sequences to be binding at different temperatures. And so this will be important ingredient for us. So if you start with that as an ingredient, you start from an alternating chain with 2 colors, and you allow this, an input for you is what are the melting temperatures that you are trying to solve over, what are the melting temperatures of these interactions. You can find solutions like this. And this is that if you start from a chain and you basically turn on yellow-yellow interaction first, you will follow just a few paths from this huge tree that end up in this geometry here. If you then allow blue-yellow to form, it will lead you here. And finally, if you allow for blue-blue to form, you will end up in this one geometry. This is completely what is known as downhill folding. It's not equilibrium. But by designing these interactions and the times when they are on and off and in sequence, you can actually get to a geometry which is not the minimum in the landscape. And so this is the crucial thing. So this sequence plus protocol is what funnels the landscape and leads you to really actually folding and making a desired structure. This was just an example. Again, working with these small structures, you can actually enumerate all the possibilities. It's not that hard. And this is what is written here. So the number of solutions you can find as a function of different system size. So blues correspond to just two species. And so this black line is the one from here. So the number of geometries. So with 2 colors, there is you can make a lot of all the possible geometries with just 2 colors and sequence protocol selection. If you introduce a 3rd color, so everyone until 11, and most of them until 13 can also be made. So this was all theoretical, but then of course it doesn't make sense unless it works with the experiment. And then Simon was saying in his first lecture something that also my professors taught me, although they were a little bit more brutal. They would tell me, experiment is a place where theory comes to die. So you can say whatever you want, but if it doesn't work with experiments, it's not a good theory. So we basically were working with Jasna Brujic's lab at NYU, where they make what are known as colloidomers. They can chain alternate sequences of basically chains of droplets. So by designing, this is sort of a recipe for the design of different interactions. Oh, sorry. And then if you just look at this is just a part of a huge view field of the experiment. This is statistics of a chain size. And you can see it's roughly exponential. So they can reach in a really short amount of time, and they can get reasonable amount of large polymers.
It's colloid or emulsion?
These are emulsion droplets coated with DNA.
You can do the same story with real colloids, but you have to put a lipid bilayer on top. Otherwise, you don't have the mobility of the DNA. If you don't do that, you can still coat the colloids with DNA. The issue that you then have is the rolling. And this is a pain. And so Ben Rogers has a recipe that works, but I think he's the only one who has a recipe that works. So unless you work with him, you don't have the recipe. So I think it's a huge issue. Emulsions or colloids with lipid bilayer just work like a charm. Yes.
Confused.
Yes.
But do you take into account if the interaction AA-BB is stronger than AB or vice versa with this activation?
Yes, exactly. So this is
what you do in the design process. You say, okay, so what are the options I can have? In this case, it's simple. It doesn't have to be even tuned. You can say, okay, so let me see if I have a protocol that is first yellow, yellow on, then blue, yellow on, then blue, blue on. How is my tree pruned, and what do I get? I can also do, what if I have blue, blue, and yellow, yellow on at the same time, or they have the same strength, and then I turn on blue, yellow, what happens? So there is no, we are not solving the inverse problem here because it's a simple enough problem that you could just know all the possibilities. You can enumerate everything. You don't have to solve the inverse problem. Okay, does that answer your question? Okay, good. Okay, so this is an example. So we give our predictions and then hold our breath. And then this is an example of a success. We're starting from high temperature in steps of 10, 15 minutes. So Angus would change the temperature, which then triggers binding of certain color pairs. And this particular protocol leads to folding of our crowning achievement, yeah, unintended. Okay, so it really works. It's really robust. And if you take just two colors, and we go back to what I showed you before, even whether or not you're assembling from a gas or in 2D or 3D, if you just have two colors, the best you can do is there is one structure of 8 that you can do with two colors. Otherwise, you just get crystals. You cannot get an arbitrary shaped function with just two colors. But if you put in a sequence, if you put in extra information in your system by chaining the particles in a specific way, you can actually get other structures. And just for fun, because I've been showing you these trees, this is basically from experiments, the resulting tree. So where you see experimental picture, these are the states that have been visited in the experiments, the ones that were not visited. And then a nice thing about this, sort of you get sort of a error correction for free in these 2D experiments, because there are two local minima in the low energy local minima in the tree, and in simulation this tells us that your yield should be lower. But these 2D experiments are sort of quasi 2D because they do index and density matching, so they're sort of sitting in a dish somewhere in the middle. So they have actually access to the third dimension. So if you let them like that for a very long time, they will eventually be some movement. But on the shorter time scale, some rearrangements are actually possible. So what happened here is that these local minima, which in 2 dimensions, if you're strictly in 2 dimensions, you can only get out of if you break a bond. In 3 dimensions, if you just flip a triangle, you can get out of them. And this is what happens. So the sort of the yield is 100% these things in experiments. And this is just a yield as a function of time, counting different structures in a row, they're color matched. So you can see how at some point we have these local minima that we thought are going to be detrimental, but actually they sort of fix themselves. If you take another design, like this one here, where you just start with yellow, yellow. So this one is just one interaction, blue, blue on, and you get this structure. If you have just yellow, yellow, basically from the experiments, you visit these states and end up in this floppy structure. And then depending on what you do next, you can have different geometries. And so this is sort of a hall of fame from the experiments. And these are what we call foldamers. So it will be important for the next part of the talk. So again, these are colloidal chains that now folded into a Lego-like pieces. So these are now new building blocks that have arrangements of colors on their surface in a specific way, and they have certain geometry, right? So you can imagine, so we call them foldamers, and now they're going to be our building blocks for the next stage. But before we do that, so it's not, this is simulation, this is experiment, but they're so beautiful, they always seem like they're better than simulation. But you can see that it's really, it works quite nicely. And the yields are decent, even with just 2 colors. Okay, yes?
Well, I mean, they remain in the same shape, or if you turn up the temperature again, they change.
Okay, very good. So this was all done in the downhill folding regime. It means that you just decrease temperature and then keep the system at that temperature, in which case it will just stay in this state. If I now increase the temperature again, I will just melt all the bonds, reach the chain state again, and I can repeat the process. So they're quite stable. To my understanding is that they can use them for a month to gather statistics without any issues with the emulsion droplets or the deterioration of DNA. Okay, so we sort of, well, hopefully you have an idea of how you can assemble things from a gas, how you can do that if you are going from chains. But now the fun part is, of course, using these foldamers as building blocks to do something else. And so now a question: how many of you know what space invaders are? Raise your hand. It's too many people. Okay, usually it's all these again that raise their hand. So to me, when I saw this, it looks like a space invader. Anyways, so it's we can make these different foldamers, and then we can ask a question: so how many of them are capable of building self-limited assemblies? Self-limited means that they pick a size, pick a shape that stays like that. And this is an example of like a fiber-like structure. So once you make this, I forgot to draw the sequence. So this is not an alternate sequence anymore. It's a specific sequence. And then you turn on just blue likes blue, which ends up in this shape. But then what they can do in experiment, and this is what they're trying to do now, is they can make these blue have 2 degrees of freedom. So blue likes blue, but it likes it at different temperatures as well. So you can make sorry, you can make them assemble these shapes and then come together and start building fibers. In this case, it's just red likes red. So you make the shape by turning on, playing with blue and yellow, and then if you just put them together and say red likes red, you will just end up with these small complexes. So all of a sudden we have this range of things we can do. Again, thinking about what nature does and protein complexes that we see in nature. But so this gives us a lot of inspiration, and then that led us to thinking about shells. They are sort of notoriously difficult to make in these self-assembling systems. You have to have various kinds of tricks, and then very recently there was this paper from Seth Freyden and Henrik Dietz's lab, where these triangular pieces that you see are just the DNA tiles, so basically constructs made out of DNA that have then very specific interactions on the facets. And then basically what they do is when they put them in a solution, they will, because they have also angles and specific interactions, they will come together and form these shell-like objects that are actually capable of trapping things. So this was post-COVID, so it was a big hype because they were proposing things that can be used to trap viruses. More recently, Peter Schultz, Francesco Shortino, and John Russo did a huge simulational study using their set assembly design tool and showed how you can, if you had patchy particles, you can actually, depending on how many species you have, how successfully you can actually build shells of different sizes. Now, we don't have patches, our particles are still these very simple ones, spherical, and then we have mobile DNA strands that go around. So we were trying to figure out if we can actually make a shell-like geometry just from folding a chain of a certain sequence. So icosahedral shells means we need a chain of length of 12. The best we can do, or find a solution with just 2 colors, is a sequence of 10. And you end up with a shell-like structure, but it's not an icosahedron. And so if you just look at the number of geometries as a function of system size for both in 2 and 3D, for 2 and 3 colors. So in 3D, you have nothing, no solutions for size 12. And in 2D, the solutions that you have for size 12, none of them are capable of making an icosahedron. But what we do have is a lot of solutions of size 6, 7, and 8. So we started thinking, okay, so maybe instead of making things out of 12 immediately, can we use our foldomers, our already formed building blocks, and use them now as in the next step, just as a next step level building block, so supercolloidal building block, that then comes together and forms a net that then is capable of trapping objects or folding into an icosahedron. And so this is what we started doing. Now I'll go very quickly through it because this is just to show. So again, we are dealing with these trees, these assembly trees, how and which path is selected, and its own non-equilibrium is very, very important. And basically for different combinations and different solutions, we try to evaluate how good the whole approach, the whole protocol is. So the ingredients for this basically evaluation are, how careful we have to control sequence because that makes experiments easy or very difficult. Do we have to do something with the bonds at different stages? Do we have to freeze, what is known freezing the bonds, basically make them inert at some stages and then active in others? Do we have to have valence control? Can you just put everything in one pot and forget about the whole thing? In 2D chirality, especially very painful. So how things fold really matters. So do we have to have a certain chirality to get things if you look at the trees, do the trees have only icosahedral nets or they have something else? How many steps do we have in a protocol? We can just turn on all the interactions at once, or we have to do it in steps? How many local minima we have that can impact the final yield? And finally, bond strength optimization. It's a very long list. Students don't like us because it's a lot of work to figure out all these things. But then once you have it, then we look at the assembly and the folding speed and really yield of these structures. So this is just a Hall of Fame, again, one that now in experiments they are testing, which is super nice. What I'm just going to show you is simulations. Okay. They're just different examples where two foldamers come together, form a net, and then this is the slowest one, and then eventually can trap objects. And again, these are the ones that are the easiest because it's alternate sequence, the one where they already have the system working in the lab. So we are very hopeful that this will work. So we are waiting for results. But so far, what we're trying to understand is how good certain strategies are. And then we do that by analyzing what I showed, this list of features, and then also assembly time and yield. And there are clearly ones that are much better than others. Okay, so we know we understand how one can assemble things from a gas, from a chain, how you can, once you have these certain size building blocks folded, how you can use those now as building blocks to make something else. Then, of course, one of the main things in this artificial life community people are obsessed about, including me, is how you can make things replicate and how difficult it is actually to replicate things. And then more recently, we realized, okay, we cannot look at replication without understanding how we can actually really build catalysts. So the last few minutes I will spend on talking about, giving you an example of how you can do this in artificial systems. And again, it all boils down to the design of interactions between building blocks and understanding the geometry of the object that you are building. Okay. So one of the most impactful work on me and many others who are interested in replication was work by von Neumann. He wrote about the theory of self-replication. Like he was focusing on cellular automata, and his position was, okay, I'm not going to care about how nature does it. I'm just going to say, okay, I want to replicate I want to make a process or a structure that replicates. Then I want to understand what are the ingredients that I need to do it. Again, without looking at any constraints from physics or chemistry. And so that's how he made his famous 32-state cellular automata that can actually replicate. And since then, there was a lot of years that nothing happened. And then at the early 2000s, well, actually, this is 2018. So the last 20 years or so, experiments, I mean, lots of work was computational, but the experiments actually started coming up in the last 20 years, where people managed, again, with these DNA constructs. So this was the key ingredient really that opened up the field dramatically. They were able to design structures where they can actually measure replication. So this is from Paul Chaikin's lab. So what he basically does is take these cross-like tiles, DNA tiles, that he makes a dimer of, and these are the replicators that he then immerses in a sea of, again, floating cross tiles. And then by playing with light and temperature, what happens is that these cross tiles bind on either side of these cross pair. And then in the cycle when the temperature is higher, the things separate from the the parent, and then can also make copies themselves. They can observe exponential growth. This is another example where it's not a direct replication, but sort of in hypercyclic ones. So Turing structure makes a bigger ring that then expels parts that can make a smaller ring. So it's sort of a little bit convoluted, but in the end, you actually have an exponential growth of both rings. This is an older paper from Rebecca Schulman's lab, where it's not exactly, they call it self-replication. It's not exactly a replication, but they have a nucleation of a specific sequence in a growing DNA type crystal. And so this was really changed a lot of what we think is possible, because for a long time it was thought that we can never have anything that can replicate. So we did something similar with again our clusters, because those are the ones that we know how they work. And so this is an example of a 3D structure. Well, actually, two parents that are immersed in a solution of particles following certain interaction rules that I will show. These particles then can bind on the surface of both parents. Everything, it's just a one-pot thing. Everything interacts following certain interaction rules. And at some point, particles on both surfaces of two parents can come together, and by following these interaction rules, interact, separate from the parents, and make a copy of both the original structure and the second parent structure. And so this is all that you need, well, all under quotation. It's not simple by far, but it's a set of interactions between different building blocks. And what are crucial ingredients is what is called the particle species valence that I told you about, that exists in experiments, and it works, it's very robust. But another thing that we need is what we call the time-dependent interactions. So this somehow has to separate from the parents. And if you design these DNAs, which is possible in experiments that have a certain, so that the bonds have a certain lifetime, this can work. So this is just from a simulation where we also measure exponential growth. I think not much can be, I don't know how much can be seen. Eventually it grows, but so what I will show you is just a 2D example, because it's a simpler one to see. So this is a template. Here it's only one parent. For some geometries it can be more, one parent. For some it's not possible. But basically what happens is that particles following some interaction rules glued to the surface of the parent, eventually the chain that is being replicated is formed. Eventually it's separated, and now it's basically folding into a copy. Eventually it will fold. Okay. Do we wait for it to fold or not? That's the question. Okay, it folded. So you can get a copy. So this whole separation business is really a challenge. And then what we are trying to understand is when is copying, from 1D like the DNA does, or 2D like I just showed you, or 3D, whether there are advantages or disadvantages, or whether or not what nature selected is really the best solution. We don't have answers to that. But then what we also realized with this separation, because again we were just tuning interactions, but it's a challenge for experiments. So it's not easily done in experiments, this whole setup. So what we were trying to understand is, okay, maybe we really should understand how catalysis works before doing autocatalysis. And so we now change stories a little bit, because we're going to talk briefly about catalysis. This is one of the most, so the next two plots that I show, this one included, are one of the most mesmerizing things that I think you can see, at least I can see when I look at nature. So it's you have a time scale of different reactions. These are the reactions we're talking about, bond cleaving of CCPO, CHCO, and bond. So these are the times, but then there is also rates of these different, on the same plot, the rates of these different reactions. And you can actually see that all these things are incredibly long, right? And yet we are alive and things function in us and everything is nice and peachy. And the reason it's like that is because of enzymes. So what you see here are these same reactions, their rate constants without enzymes. And then whoop, if you put enzymes, if you have something to speed up the process, they all end up on here. So here, ADC, I forget which one it is, it goes 17 orders of magnitude, right? With the presence of an enzyme. So really, everything we are in it, everything is because we have enzymes, we have catalysts that can cleave or make bonds very, very efficiently. And yeah, so I don't know. For me, this is like mind blowing. So we are obsessed with enzymes. We really, I mean, we cannot make an enzyme, but we at least can try to understand how you can make artificial catalysts. Now, yeah, so it's really the crux of the challenge is on the theoretical side. Okay, so there is no framework for designing catalysts. So what this is the standard story from textbooks that you are familiar with. So you have a reaction, that you have a substrate and a product, there's a transition state. What catalyst does, it somehow comes, right, and reduces this barrier and makes the reaction more feasible. But so if you again, you look at the nature, it is the table that I showed you. You have this huge range of enzymes that do amazing things. But now if you look at the industry, we are not that successful, right? Our industrial catalysts can speed up things, but not 17 order of magnitude. Nothing happens 17 orders of magnitude. It's a few orders of magnitude that you hope for if it speeds up that much. And there is a lot of work going on in like David Baker's lab in Washington, where they're trying to really build something, some new like catalyst, new enzymes. And there was also a Nobel Prize, right, in directed evolution a couple of years back with Francis Ertoldi. So they're trying to use these different methods where they start from existing things and then change or a little bit and then try to make new ones. But it really, we don't know how to make from scratch catalysts. Okay, and the main problem is sort of, one of the main problems is summarized in this plot here, which is known as Sabatier principle. Maybe in chemistry, if you had chemistry, this is what you've seen in your studies. And this is that, so you have a substrate, right? It needs to bind to a catalyst. A reaction has to happen and it has to detach. So on one side, it has to bind strongly for a reaction to happen. But then on the other side, it has to detach quickly so that the reaction is very efficient, right? So there is a problem right there, and this is what's known as a Sabatier principle. So there is some optimum bond strength with which things attach to the catalyst, and then the reaction works. And then the key element here is what is called a transition state, and Pauling was one who proposed that it's not only about this that the substrate that the catalyst binds substrate strongly, but it is that it's actually binding strongly the transition state, so something slightly different. And so again, a lot of theory and sort of, in some sense, experiments with limited success over the last decades. And what people do is either focusing on just this, what is called the transition state theory from Pauling principles, or they're doing these all-atom molecular dynamic simulations in which they maybe modify something in the enzyme, then try to see if it makes a better catalyst or not. Okay, so we are not doing anything, none of this. So we're just going to try to, again, with the ingredients that we know and have, try to build a catalyst to see if we can do that, focusing on bond cleaving. So we will have a substrate that spontaneously can make a product. It's very hard to do that. But if it attaches to a catalyst, through a set of reactions, we can actually reach the product. So basically what we are doing, remember I showed you all these different trees, assembly trees and folding trees and all kinds of trees. None of those pathways are good enough for the reaction that we want to see. So what we are doing, we are adding a pathway in the whole system. We are adding a pathway that then enables us actually to do a reaction much faster. So this is again just briefly. It's a very simple catalyst. It's a dimer that is capable of breaking a bond. So this is a spontaneous reaction. So a bond spontaneously breaks to form a product. It's a very, very, very strong bond. So the time scale of simulations, you don't see this event happening. And so the idea is, okay, so now what is the additional pathway? We take our dimer, it attaches to our catalyst. One particle, eventually the second particle is also attached. This causes a reaction, which leads to breaking of a bond, and I will explain what the reaction is. And then eventually these monomers separate individually from the catalyst to make a product. This is all quasi one-dimensional. Basically what happens is that, so if this geometry of the catalyst, is basically distance between these two particles, is not the right one, if the substrate attaches, it will find, so this is sort of two reaction coordinates, which are distances of these particles with respect to the catalyst. So what will happen if they attach, they will find the minimum position and make a structure very stable. But if the geometry is just right, meaning that the distance between these catalyst particles is large enough, when the substrate attaches, when it's trying to attach to the second particle, the bond between the substrate particles is being strained. And so it can never find a minimum, some stable position, but it actually just immediately breaks and the bond is broken. And so this is just an example. It's a sped up simulation. So this is a spontaneous reaction system. So this is a reference system. This is the one where you have a few of these catalysts floating around. And eventually, what you will start seeing are green particles, which are the monomers that are being separated from the catalyst. There is no energy input here. Okay, so this is a crucial thing to understand. So it's completely all interaction design. So there is we are cycling nothing. We're not putting any energy. It's all interaction and geometry of the catalyst design. So we made the geometry such that we are straining the bond as the substrate is trying to attach completely to the catalyst. And then eventually, things spontaneously separate. So this is just an example. This is sort of for a specific set of parameters. This is how strongly you need to bind to the catalyst versus the bond that you're trying to break. And so this is the main thing, right? So the bond can be as strong as you want, and you don't have to bind to a catalyst as strongly to actually break it. So you want to be where this yellow, the sweet spot is. All the simulations that I showed you again are calibrated to experiments. So it's really something that experimentally is feasible, and hopefully our colleagues will do that. And this is just a summary of concentration of these monomers as a function of time. And this is the reference system. And this is the system with a catalyst present. So it's really catalyst that works. But what the difference compared to what was before is that we really understand how the geometry plays into the whole story and what is it that we need to optimize for and what are really the crucial points in this whole additional pathway that we are adding to the reaction. Okay, and with that, I think I covered most of the things. So hopefully, if there is one thing that you remember from the whole talk is, okay, so if I want to assemble this from a set of colloids, I can just arrange them, take who likes who, the nearest neighbors, I only like my nearest neighbors and no one else, and I can assemble it in a solution. That's a recipe that works. But also, I think hopefully I gave you a bit of a flavor of what you can do if you understand the geometry and the interaction design between specific building blocks to achieve bio-inspired processes and structures. And of course, nothing can happen without actually other people doing stuff. So this is my PhD student, Natalie. So I showed you a little bit of her work connected with shells. Most of the work on folding was done by my previous PhD student, Maitane, who is now here at ISTA. And then experiments and some theories done in Jasna Brujic's lab by Rabea and Angus, who was then a postdoc. Then some other people are doing experiments right now. With that, I'd like to thank you, and I'm happy to take more questions.